Welcome to Computational Genome Biology. Today, the way we treat cancer is changing. We are moving away from categorizing tumors strictly by the organs they originate in, and instead targeting the specific genetic errors driving them. But what happens when standard genetic tests are blind to the mutation powering a deadly tumor? To treat cancer effectively today, we have to map its precise genetic engine, rather than just locating its physical presence in the body. Traditionally, molecular diagnostics relied on sequential single-gene tests or small targeted sequencing panels. These targeted panels scan a highly curated set of genes, searching for known hotspot mutations, but they only look at less than 1.5 megabases of data out of the entire genome. They provide a deep look, but at a very narrow target. Inside the body, tumors are characterized by intra-tumor heterogeneity. This means a single tumor is actually made up of highly diverse, constantly mutating subpopulations of cells. Because small panels only illuminate predefined areas, they are completely blind to rare fusion events and emerging biomarkers lurking in these diverse populations. Relying solely on targeted panels forces oncologists into sequential testing, which depletes valuable biopsy tissue, delays diagnosis, and ultimately causes patients to miss out on immunotherapies. To solve the blind spots of targeted panels, clinical practice has evolved toward whole exome sequencing, or WES. We can compare WES to whole genome sequencing, which scans a patient's entire genetic makeup. Whole genome sequencing produces an enormous amount of data, making it too computationally heavy and expensive for routine clinical care. Whole exome sequencing, instead, targets only the protein coding regions of the human genome. This region makes up only about 1 to 2% of our total DNA, yet that tiny fraction houses roughly 85% of all known disease-causing variants. This places whole exome sequencing in a diagnostic sweet spot, providing maximum actionable clinical data without the computationally crippling bloat of full genome sequencing. This broad data capture allows oncologists to accurately calculate a complex signature known as tumor mutational burden, or TMB, which helps determine if a patient will respond to immunotherapy. This chart compares the accuracy and data spread of TMB calculation between targeted sequencing panels and whole exome sequencing. There is a 10 to 15% discordance rate between the two methods, meaning small panels frequently miscalculate TMB and miscategorize a patient's immunotherapy eligibility. Whole exome sequencing is also utilized as a repeat sequencing tool when a patient develops drug resistance. Under the pressure of targeted therapies, tumors undergo subclonal clonal evolution. They splinter and select for novel, resilient mutations that can evade the current treatment. Repeat whole exome sequencing acts as a real-time tracking system. It allows oncologists to monitor the emergence of these resistant driver mutations and adjust targeted therapy combinations immediately. The integration of whole exome sequencing has transitioned from an experimental research tool to an integral part of routine oncology. Real-world data validates this transition. For patients matched to targeted therapies using exome scale profiling, this graph shows a 31% longer overall survival rate. It also resolves the diagnostic origin for 63% of cancer of unknown primary cases. To support this level of testing, the Centers for Medicare and Medicaid Services established National Coverage Determination 90.2. This provides national coverage for diagnostic next-generation sequencing assays in patients with advanced, recurrent, or refractory malignancies. Additionally, the FDA has approved assays like Illumina's TrueSight Oncology, authorizing specific CPT codes that make comprehensive testing financially sustainable for clinical laboratories. Robust reimbursement and regulatory pathways are the true engines that move a technology from the lab bench to the patient's bedside. To optimize this technology, oncology networks are pushing for universal, proactive reflex testing. This means triggering an automated exome sequencing protocol at the initial diagnosis of an advanced solid tumor, rather than waiting for sequential orders. Laboratories are also transitioning to unified dual DNA and RNA workflows. This conserves highly valuable biopsy tissue while maximizing the diagnostic yield for targetable fusion events. By identifying the exact molecular drivers of aggressive malignancies up front, whole exome sequencing is replacing empiric palliative chemotherapy with targeted, tumor-type specific treatment strategies. Thank you for watching Computational Genome Biology. If you found this helpful, please like and subscribe for more deep dives into the science of genomics.